And so I'm going to be going off of the assumption that we sort of have a little bit of a prior understanding of some of the fundamental concepts behind these processes. So such as DNA replication and a little bit of the overview of the transcription translation process as well. So some of these topics are going to be discussed briefly, and then others we're going to go into more detail. So transcription and translation are going to be covered in more detail as well. And so just a little bit of an overview. During transcription, the double-stranded DNA separates and RNA polymerase begins moving in the 5' to 3' direction. And so as RNA polymerase is moving along that single strand of DNA, a strand of mRNA is going to be synthesized. And it's important to note here that the direct product of transcription in eukaryotes is actually going to be what's referred to as pre-mRNA, and that's the unedited form of mRNA. And so this pre-mRNA is going to contain exons, which are the coding regions, and it's also going to contain introns, which are the non-coding regions. And then when the introns, again, those are the non-coding regions, are spliced out, that's going to be what we refer to as the mature form of mRNA, which results after. And that mRNA product is going to be complementary and anti-parallel to the DNA template strand. mRNA is then going to go through what's known as translation to become a protein. So this step occurs when ribosomes are recruited to the mRNA strand, and then they begin synthesizing the protein in that 5' to 3' direction. That's going to produce a protein that begins at the amino terminus or the N terminus, and it's going to end at the carboxy terminus, which is also called the C terminus. And so there are 3 main types of RNA, and those are going to be mRNA, which is messenger RNA, tRNA, which is known as transfer RNA, and rRNA, which is ribosomal RNA. And there are other forms of RNA as well that are more specialized. We're going to cover those in more detail as we go on. So the first one is messenger RNA, and messenger RNA is crucial in making sure that the genetic information that's on that DNA template strand gets accurately turned into the corresponding amino acid sequence. And so this is kind of important for the production of a functional protein, because it's important that that information gets relayed in the way that it's intended to for that specific protein to be produced. However, after the mRNA is synthesized, which occurs in the nucleus, it can undergo a series of what's known as post-transcriptional modifications. Those post-transcriptional modifications, as the name applies, are going to be after transcription occurs. And mRNA is the only type of RNA that contains information that then gets translated into proteins. And as such, it has a specific language that, quote unquote, translates the instructions from the DNA to the protein. The strand of mRNA, as such, it has a specific language that then translates those instructions from DNA to protein. The strand of mRNA is read in 3 nucleotide segments, and these are what are referred to as codons. Codons either instruct the cell to place a specific amino acid along the sequence, or they can act as a cell signal, so like a stop codon, during the protein synthesis process. That stop codon is just basically going to halt transcription. It's also important to note that in eukaryotes, mRNA is going to be what we call monocistronic, and that's when this means only one protein product is going to be produced per mRNA molecule. In prokaryotes, however, that mRNA can be what's known as polycistronic, and that's going to be when multiple proteins can be produced depending on where along the strand of mRNA translation begins. And so the next type of RNA is what's called transfer RNA or tRNA. And transfer RNA is responsible for converting the language of nucleic acids into the language of amino acids, like we said before. It's going to contain a folded strand of RNA, and that's going to include a 3-nucleotide anticodon. And so this anticodon is going to be complementary to the codon on the mRNA. The anticodon then is going to recognize and pair with the appropriate codon on the mRNA, and that's going to be while they're all part of that ribosome complex. And so there are a total of 20 amino acids in eukaryotes, and at least one codon is going to be responsible for representing each of these amino acids. And when that tRNA then picks up the corresponding amino acid, it's going to be what we refer to as charged or activated. It's also important to note that mature tRNA is going to be found in the cytoplasm of the cell. And so this is just the structure of the tRNA molecule. And this highlighted region at the bottom is going to be the anticodon sequence. And this is going to be complementary to the codon on the mRNA molecule. And the 3' end is going to be where the amino acid would be attached when the tRNA is in its charged or activated state. So that's going to happen up here at the 3' end of this molecule. And each type of amino acid has its own corresponding amino acyl tRNA synthetase. That synthetase molecule basically just facilitates the transfer of the activated amino acid complex to the 3' end of the correct tRNA molecule. Because that's an energy-rich process, it's going to require 2 high-energy bonds that will be from ATP. It's also important just to note that in its activated state, the tRNA molecule is what's referred to as amino acyl tRNA. And so this is just a picture that kind of diagrams the complex together. Here we're going to see both the mRNA and the tRNA meeting at the ribosome complex. So this would be probably when translation begins to occur over here. And the third form of RNA is what's known as ribosomal RNA, or rRNA. And rRNA is synthesized in the nucleus, also functions as an integral part of the ribosomal machinery that's used during the protein assembly process, and that occurs in the cytoplasm. So many rRNA molecules can function as what's known as a ribozyme. And ribozymes are just sort of enzymes that are made up of RNA molecules instead of peptides, proteins, like we would think of an enzyme being. And as that name implies, ribozyme, they're going to have catalytic activity. So they're going to be able to play a part in things like splicing and gene expression later on.